Hello everyone. So welcome to our new lecture. Now in this lecture we are going to see another useful microorganism, that we are studying in this chapter number 8, useful and harmful microbes. In our previous lecture we have studied one of the useful microorganism, that was rhizobium. So we know that this particular bacteria is a pinkish rod shaped organism which is found in root nodules of leguminous plants, and it is used to supply nitrates, nitrites, and amino acids to the plant. Okay, so this useful microorganism, which is rhizobium, is very much helpful for the good growth of plants. Okay, now let us see another useful microorganism, and that is a yeast. Okay, so before going to the role and importance of this particular microorganism, let us see one of the experiment which is used to understand this microbe. So the first step in the experiment is mix active dry yeast, sugar, and lukewarm water in a bottle. Fix a colorless transparent balloon on the mouth of the bottle. So what is the first step, or what is the first procedure in this experiment? We have to take an active dry yeast. Okay, we have to buy that yeast from the market. Then we are going to add that yeast with sugar in a lukewarm water. Okay, so the water which is not completely boiled, okay, just heated. In that water we are going to add this yeast. Okay, and after that we will be fixing the mouth of the bottle in which we have put all this yeast, sugar, and water. Okay, we are going to fix that mouth with a colorless transparent balloon. Okay, now after some time what we will observe, that balloon is going to inflate. Okay, and a gas is going to be collected in the balloon. So the next procedure is we have to mix that gas with the lime water, and let us see what will happen if we mix the gas in the lime water. Okay, this will be our first observation. Then we have to take that lime water, okay, collect that lime water in a beaker, and we have to observe it. Then another procedure is take a drop of solution from the bottle on a glass slide, put a cover slip, and observe it. So in this experiment we have to observe two different procedures, okay, and we have to try to understand our observations. So first observation will be, now as we are going to add the gas which is accumulated in the balloon, okay, due to yeast which is mixed with sugar solution. Okay, so that gas will change the lime water. Okay, it will turn lime water milky. That means that the gas which is generated in the water bottle, or which is collected in the balloon, is a कार्बन डाइऑक्साइड गैस. ओके, जब हम लोग ड्राई ईस्ट को शुगर में ऐड करेंगे और उसे ल्यूक वार्म वाटर में ऐड करके बॉटल में डाल देंगे, तो क्या होगा, एक गैस रिलीज होगा. और वो जो गैस है वो कौन सा गैस रिलीज होने वाला है, कार्बन डाइऑक्साइड गैस रिलीज होने वाला है. तो जैसे ही हम लोग CO2 को लाइम वाटर में ऐड करेंगे, हमारा ऑब्जर्वेशन क्या होगा कि जो लाइम वाटर है उसका कलर चेंज होगा, इट विल टर्न मिल्की. ओके, सो अगर लाइम वाटर मिल्की हो रहा है, मिल्की टर्न हो रहा है, वो इसका मतलब क्या है कि CO2 गैस जनरेट हुआ है. ओके, क्योंकि कार्बन डाइऑक्साइड गैस जो है वो लाइम वाटर को क्या करता है, उसके ऊपर उसका रिएक्शन होता है और लाइम वाटर का कलर चेंज होता है. In the second observation, when we are going to take the solution from the bottle on a glass slide, okay, drop off the solution, and when we will observe that slide under a microscope, we will be looking at a colorless unicellular oval shaped fungus. Okay, it will be a colorless unicellular oval shaped fungus. So yeast, we are going to observe yeast, okay, under the microscope. Now yeast grows and multiplies quickly due to carbon compounds in the sugar solution. Now we know the molecular formula of sugar, that is C2L H22 O11. So this is the molecular formula of sugar. Now definitely you know in this compound carbon is there and hydrogen is there. That means it is a carbohydrate. Okay, so this carbohydrate is treated by the yeast, okay, and it is converted into a carbon dioxide and alcohol, okay. But due to this carbon compound, the yeast is going to grow quickly, okay. The reproduction between the yeast molecules, between the yeast microbes is going to be very much quick, okay. Now the process where yeast converts the carbohydrates into alcohol and carbon dioxide, this process is called as fermentation. What it is called? It is called as fermentation. Okay, so this is the process which is nothing but a fermentation of the sugar solution. Now as we know that yeast is a colorless unicellular oval shaped fungus, so definitely it belongs to the kingdom fungi. And there are almost 1500 different species of yeast. Okay, how much species are there in existence? Almost 1500 different species are there of this particular microbe. Now yeast is nothing but a eukaryotic cell, okay, unicellular organism but eukaryotic. Okay, what is the structure of this yeast cell? It is eukaryotic in nature. Okay, now this is the structure or the physical parameter of the yeast. Now let us see the application of yeast. Okay, now molasses produced while extracting sugarcane juice are fermented with the help of yeast Saccharomyces to produce ethanol, that is primary product, and other alcohols and ester as a secondary product. Okay, so we all know that with the help of sugarcane, we produce sugar. Okay, so in the sugar factory, along with the sugar, alcohol is also produced. And that alcohol is produced by fermenting the molasses, okay, the byproduct of the sugarcane. Okay, and to ferment that molasses, one of the yeast, that is Saccharomyces, is used, okay, and this yeast Saccharomyces converts the molasses into ethanol, okay, and other alcohols along with the ester. So this is the first application that Saccharomyces can be used to produce ethanol. Okay, now ethanol is very much useful to obtain spirit and other alcohols. So with the help of ethanol, which is produced, okay, with the help of yeast, that can be used to make spirit and other alcohols. Okay, and ethanol is a very much useful product. Why it is useful? Because it is smokeless and high quality fuel. Okay, so we can use ethanol as a fuel because it is smokeless. It does not emits any kind of harmful gas, okay, after burning. So it is very useful as a fuel. So do you know that in every fuel that we use, that is petrol and diesel, 10% of ethanol is mixed, okay. And the reason behind mixing this 10% ethanol in this fuel is that ethanol reduces the amount of pollution caused by those different fossil fuels, okay, and it also increases the efficiency of the fuel. So it is very much useful to us. Then glucose and fructose, that is the sugars present in the grape juice, are also fermented with the help of yeast to produce the alcohol which is used for making wines. Okay, so grape, okay, or grape juice, in that also we will find some sugar, that is glucose and fructose. Again, these are nothing but the carbohydrates. Okay, so glucose, that is C6H12O6, okay, and fructose C6H12O5, both are the carbohydrates, and these carbohydrates are again fermented with the help of yeast, okay, and what we are going to get? We are going to get an alcohol, and that alcohol is used for making wine. Okay, it is used for making wine. So this is the application of this particular fungi or microbe, that is yeast. Okay, another application is that yeast is used to make breads. Okay, breads are made with the help of yeast. Okay, so what it does, it makes the bread spongy and puffy. So this was the another application of yeast. Now let us see one more application which is categorized as bioremediation. So what is bioremediation? It is nothing but a process wherein we kill the pollutants or other harmful bacteria and microbes with the help of another microbe. Okay, that is nothing but bioremediation. So if we are reducing the pollutant from the environment or from a particular substance, or we are eliminating the harmful microorganism which is there present in the environment with the help of another microorganism, then that process is nothing but bioremediation. So yeast, which is Yerovia lipolytica, okay, it is used to produce or to absorb the toxins, it is used to absorb the toxins released during the production of palm oil and the heavy metals and the minerals released in some other industrial processes. So whatever harmful substances are produced in the production of palm oil and while treating the heavy metals and minerals, all those harmful toxins, harmful substances can be eliminated, can be eradicated with the help of this particular yeast, which is called as Yerovia lipolytica. Okay, so this is the first application considering the bioremediation. Then second, Saccharomyces cerevisiae, okay, is used to absorb arsenic. So again, one of the harmful pollutant, which is arsenic, which is the major pollutant found in soil. Okay, this can be eliminated with the help of Saccharomyces cerevisiae. Okay, then third application, oil spills in oceans are cleaned with the help of alkali borax bacteria. So the oil spills which may take place due to some accidental activity, okay, in the seas and oceans, and those oil spills can be cleaned with the help of bacteria, and that bacteria is alkali borax. Okay, it is not a fungi and it not belong to the yeast category, but this bacteria is used to clean the oil spillage. Okay, so these are the application of yeast. Now in this lecture we have studied about yeast and their applications. Now from the next lecture onward we are going to study about antibiotics and some harmful microorganisms. For this lecture your homework will be, you have to draw the diagram of yeast. Okay, so the diagram which was given here, this oval shaped yeast diagram, you have to draw this particular diagram in your notebook. Okay, you can see cytoplasm, nucleus, cell membrane, chitin cell wall, mitochondria and vacuole. These are all the organelles of the yeast cell. Okay, so this is your homework. We will meet in the next lecture. Thank you.